DNA replicate by using semi-conservative methods that results in the formation of double-stranded DNA with one parental and one daughter strand. Hi there future learners, before we dive into this amazing educational content, I have a quick request for you. If you enjoy discovering new ideas and expanding your knowledge, make sure to hit that subscribe button right now. By subscribing, you will never miss out on our latest videos that are designed to inspire and educate you. So go ahead, click that subscribe button and be a part of our growing family of curious minds. Your support helps us to create more incredible content just for you. Now just look at what is DNA replication. DNA replication is a process in which each strand of DNA molecule acts as a template for the synthesis of new complementary strand. The newly synthesized DNA strand transferred to two different daughter cells. Replication occurs before a cell divides to ensure that both cells receive an exact copy of parent's genetic material. DNA replication studied in a model organism, that is the E. coli. Replication is bidirectional process as we know, and the originates as a single origin of replication that occur in DNA. DNA replication completes in three steps, that is the initiation, elongation and termination. In this video, we have to look at the initiation process, that is the DNA replication initiation in prokaryotes. In E. coli, the process of replication is initiated from origin of replication. The origin of replication in E. coli is called ORIC. The ORIC consists of a 245 base pair long AT-rich sequence which highly conserved in almost all prokaryotes. Mostly, two types of sequences present in this region, 3 repeats of 13 base pair called 13-mer and 5 repeats of 9 base pair called 9-mer sequences. Now, let's look at what are the proteins involved in the prokaryotic DNA replication process. First, look at the DNA A. It recognizes the ORIC sequence for the initiation of replication. DNA A recognizes the ORIC site for the initiation of replication. Then come to DNA B. It unwinds DNA double helix and it actually works as a helicase. Then what is the role of DNA C? At the origin of replication, DNA C helps helicase to recognize the site for its action. It helps the DNA B. Okay, then what is the role of DNA G? It acts as a primase. It synthesizes for new RNA primer. It synthesizes the new RNA primers. Then comes the DNA polymerase 1. It removes the RNA primer and replaces it with the newly synthesized DNA. Then DNA polymerase 3, that is the main enzyme that adds the nucleotides in 5' to 3' direction. Then helicase, it opens the DNA helix by breaking hydrogen bonds between the nitrogenous bases. Then the ligase, it seals the gap between Okazaki fragments to create one continuous DNA strand. Then what do the primase? Primase synthesize RNA primers needed for start replication. Sliding clamp helps to relieve the strain on the DNA when the unwinding by causing breaks and then resealing the DNA. What do the single-stranded binding proteins do? Single-stranded binding proteins binds to single-stranded DNA to prevent DNA from rewinding back. Then look at the replication fork. The replication fork is Y-shaped structure and is important because it facilitates polymerase to work properly. As a chromosome of bacteria is circular, two different replication forks are developed in two different directions and runs towards each other. Then we have to look at the mechanism of initiation of replication in prokaryotes. Protein A, that means DNA A proteins with ATP binds to the 9-mer sequence of ORIC. This binding allows the opening of 13-mer sequence of ORIC by bending of the DNA. DNA by proteins bind to the 13mer repeats which are recognized by the DNA-C. Once DNA-B is settled on the ORAC at the 13mer, then DNA-C protein will be released. DNA-B helps in the unwinding of DNA and generate two single strands of DNA. DNA gyrase helps in the unwinding of DNA and release the tension on the DNA strand by negative supercoiling. Tension or supercoiled DNA decreases the rate of replication. In this video we look at the proteins, what are the proteins and mechanism of DNA replication, initiation in prokaryotes. In the next video we will look at the elongation of DNA replication. Thanks for watching this video. Remember to subscribe to channel and let's embark on these educational adventures together.